my PhD candidate I supervised with Jay Kocherunchit. And basically, the project was related to Campylobacter safety in the poultry industry, but very much microbiology focused. So hopefully everyone can understand the talk. I'm sure Stanley will do a fantastic job in presenting it, but it is pretty much hardcore, I would say, applied research, but with a sort of a science spin, with the desire to try to help the poultry industry solve the problem with Campylobacter safety, which is in Australia has been a bit of a thorny issue. Like Salmonella, Campylobacter is one of those foodborne pathogens that's stuck around, and basically the case numbers haven't really been dropping off very quickly. They're maybe very slowly dropping off with better techniques, but with diagnosis and so forth. But generally speaking, a sort of persistent food safety issue within the country. Stanley's submitted his thesis and has also already published a number of papers in great journals. So.
All right. First of all, thank you everyone for having me today. My name is Stanley Chen. I'm doing my PhD under supervision of John and Jay in UTAS, and my supervisor in the CSIRO are Maria Fagan and Leslie Duffy. So my project is about Campylobacter persistence through poultry processing. And well, since it's launch time, so let me present you one of these juicy, delicious chicken, because it sounds lovely. It seems lovely and very delicious. But the problem is, if you don't cook properly, or it's undercooked, you may encounter one particular foodborne pathogen called Campylobacter. It is a gram-negative pathogen, and the optimum condition to grow is around 42 degrees Celsius, around the microaerobic condition, which is 5% oxygen and 2 to 10% carbon dioxide. And among all those species and the subspecies, C. jejuni and C. coli are mostly commonly found in humans, patients, and food sources. And the Campylobacter are mostly associated with the poultry and the related products. Once you are infected, usually the symptoms are self-limiting, involves 1 to 3 days fever, vomiting, and headache, and then follow that 3 to 7 days watery or bloody diarrhea with abdominal pain. Usually, because it's self-limiting, so it doesn't usually require hospital treatment. But if it does, then usually the 2 antibiotics involved are erythromycin and fluoroquinol. And in rare but severe cases, Guillain-Barré syndrome. And in Australia, Campylobacter are among the leading cause of human gastro diseases for a number of years, as you can see in the graph on the top line, and followed by the Salmonella. And especially in the last 5 years, the cases in Australia is increasing manner from 2015 up to 100, about a little bit more than 140 cases per 100,000 population last year. Comparing to the rest of the world, Australia is is one of the countries that have the most Campylobacter recorded cases, following New Zealand. And because Campylobacter is mostly associated with the poultry and the poultry-related products, and so it is important to understand the prevalence of Campylobacter on chicken. As you can see in the graph, that Australia is also have a greater Campylobacter prevalence compared to USA, EU, Africa, and China. And because chicken is a major commodity in the agriculture, and especially in the next few years, it's going to increase quite a lot. So controlling Campylobacter on chicken are quite important. And so a few years ago, the ministers in Australia released this document called Australian Foodborne Illness Reduction Strategy. And the Campylobacter and the Salmonella are the 2 focused foodborne pathogens in this document. So that's the ground of the and background reasons of this particular project. As you remember from one of the earlier slides, the Australia's prevalence of Campylobacter on chicken is about 84% at the end of the processing. And when we look at the beginning of the processing, as you can see, the positive Campylobacter positive samples are the same as the end of processing. Seems the current commercial processing doesn't actually eliminate Campylobacter or chicken very effective. So the question we have to, so the question that I'm looking at is just to look through the processing at various processing stages to understand why it's not effective. And the current processing is largely, the design of the current processing largely depend on the culture-based results before. And the culture-based results only allow us to isolate some foodborne pathogens and observe or record the results to see whether through certain processing it's effective to reduce the counts. Whereas in the recent years, the development of culture-independent techniques allow us to broadly look at the bacterial community on examples through different sources to understand the interactions and the distributions or potential contaminations from various sources. So that's also one of the techniques that I largely focused on. So to help the chicken industry, I sampled two different processing commercial lines. One is to keep the confidential, I refer to line A and B. So the major difference between these two lines is line A have all different processing stages separately, and whereas line B have one particular tunnel system incorporates scald, defeathering, and the plug. And so this is usually the commercial processing looks like when you got the chicken coming in and it got stung and bleed out, and then going through scalding. Usually that's temperature varies between different processing plants, but usually between 52 to 62. And after scalding, you go into the defering to remove the feathers and then remove the head, and then moving to evisceration to remove the gut and the intestines. And then you're going through inside-outside wash to remove the potential contamination from the C cup. And then following, you go into the immersion chilling. Usually it involves multiple immersion chilling tank, because usually in the commercial settings, when the chicken carcass is going through the immersion chilling, and at the end of the immersion chilling, the temperature usually reduced down to about 10 degrees. And then after that, depends on different processing plants. Some of them may involve a further air chill stage. That's usually the chicken hang in the air chilling room. And then by the end of this process, the temperature of the inside of a chicken carcass is usually around 4 degrees, and then move on to the cut up and grade and then packaging. So in my two samplings, I sampled the two lines, and the first sampling point is before scald, and then the line A I sampled after scald, line B I sampled after the one tunnel system, so which is after plucker. And in line A I also sampled a few C cup and after the evisceration, and then move on to the three stages that are quite similar between two lines: before immersion chilling, after immersion chilling, and after air chilling. And for line B, I also sampled the 30 carcasses after packaging to monitor their bacteria counts and the diversity through the shelf life. And in line, and to understand the relation between the sample and the environmental processing parameters, I also sampled a few environmental samples in line B that involve scalding water tank and immersion chilling water tank. And another difference between line A and B is line A has half an hour immersion chilling, and line B is 1.5 hours. And then in the air chilling, line B is half an hour, whereas line A is 1.5. So in the air chilling in line B, I also sampled the air samples, and I also put some swabbed sponges on the shackles to get enough air samples, and also swab the wall surface around the air chilling room. And so this is all the sampling strategy I have. And in terms of these results and analysis, I use the culture-based and the culture-independent techniques. And culture-independent techniques, I just monitor the bacterial diversity changes through the processing. And the culture-based, for line A, I recorded results of the total viable counts, Campylobacter and Salmonella. And line B, I recorded TVC, Campylobacter, and Pseudomonas. Because The reason I didn't include environmental samples in line A is the sample is I did these two different sampling over a year and 1.5 years. So just the reason to include a more intensive environmental samples in line B is based on the results. Some interesting results I found in line B or in line A. Sorry. So I will show you later. So this is the results of the culture-based results of the TVC and the Campylobacter through processing. On top of the graph, you got TVC and the Campylobacter in line A. Overall, you can see the reduction of the TVC and the Campylobacter are quite significant from before SCO to post-air chill. And that's the similar trend in line B from before SCO to post-air. It's significant reduction. Shows the effectiveness of the current commercial processing to reduce the total bacterial counts and the Campylobacter. And if you look, and when you look at the particular processing stage, like the scalding, it's effective, and the immersion chilling is effective as well, and the same in the line B. But when you look at the air chilling, which is the post-immersion chill and the post-air chill, there's no significant reductions in terms of the total TVC and the Campylobacter between line A and the line B. And especially in line B, you can see there's a statistically significant increase of Campylobacter in line B. So indicating a potential cross-contamination in air chill in some certain cases. Another interesting finding is in line A, about the Campylobacter counts between post-SCO and pre-immersion chill. That's where the evaporation defeathering happened, and there's a significant increase, and shows that on occasions you have a contamination happening when you're pulling the guts out of the chicken, because the process of the evaporation looks like this. You got one of the sterile handle going inside the chicken and then pull the intestines out. And because of different size of the chicken, you may potentially disrupt or rupture the intestines and then potentially distribute the content of the ceca onto the carcasses to cause the contamination. So from this part of results showing that scalding and immersion chilling generally are effective in reduced TVC and Campylobacter. Air chilling is not effective, and the potential contamination are the defeathering and the evaporation. And then here I present the Campylobacter prevalence through processing between line A and B in bar chart. So overall, between pre-SCO to post-air, the Campylobacter prevalence on carcasses I sampled is 100%. There's no reduction at all. It's similar to the national survey earlier, 84% before and after. But between the process, you can see there's some reductions, but it always goes back to 100%. So it shows the cross-contamination during the process, and especially in this part in line B, you got the air chill cause the potential contamination. That's aligned with the early last slide of the Campylobacter counts in line B is statistically significantly increased. And the box chart is just showing the culture independent technique, culture independent approach to look at the abundance of Campylobacter in the entire bacterial community. And although overall the abundance of Campylobacter in the entire community are quite low, you got some rare cases, rare examples goes upon to 1.2%, or like here, like in line B, 2.6%. But generally, the relative abundance of Campylobacter, the range are below 3%. So, so these results together shows the Campylobacter is a minor but persistent organisms in the community on carcasses. And then in line B, because I also include environmental samples, so one of the results here shows the Campylobacter in SCO water, immersion water, air samples through the air chill room. There's either below the detection limit or just significantly low. So shows the low prevalence of or presence of Campylobacter in the environment. And then on the other hand, you see the Pseudomonas counts, especially here the wall surface in the air chill and then the shackles through the air chill room got a significant relatively large counts, showing the potential contamination may happen in the air chill, particularly for the Pseudomonas. And so I looked back to the counts of Pseudomonas on carcasses in the air chill particular. However, I found that the counts actually shows no significant difference or below the detection limit. So that's potentially as I said, the detection of the counts on Pseudomonas are probably below the limit. And then the other interesting found is the Pseudomonas on the carcasses post air chill to the start of shelf life. That's during the packaging, and there's a significant increase in the Pseudomonas counts on carcasses, showing there will maybe potential contamination routes after the air chill through the cut up and packaging. So the results altogether here shows the aligned together as the effectiveness of SCOT and immersion chilling, and potential contamination routes are the evaporation, air chill, or post air chill steps, i.e. packaging, and the Campylobacter is usually in low presence in the environment. And here is the 16S amplicon sequencing data between two lines. And largely, if you look at the phyla level, the abundance, the four major phyla are the same. The Firmicutes, Actinobacteria, Proteobacteria, and Bacteroides. Although they are proportion depend on the different processing stage, are quite different. Largely on the carcasses, Firmicutes is the dominant phyla. And in line B, we interestingly find that the Proteobacteria that are very dominant in the end of shelf life and the wall in the surface. And as we know, the Proteobacteria usually involves a lot of foodborne pathogens as E. coli, Campylobacter, and Pseudomonas. So looking into the further detail will be interesting. And so because the results at the genus level involves quite a lot of different genuses, I chose top 10 or 10 most abundant genera at each processing stage and then put them together. So on the left hand, you have the line A, and on the right hand, you have the line B. And one key finding from line A is this Pseudomonas dominant abundance on the samples in post air chill. Like all four of these genera represent about nearly 50% of the entire bacterial community. And because the nature of the Pseudomonas and the other three genera, they are cyclotolerant and spoilage organisms, which the finding from here leads to include environmental samples in the air chill in line B. However, in the Pseudomonas, we did find significant presence of Pseudomonas on the wall of the air chill, and a little bit in the on the sponge of the shackles, and slightly in the air. Slightly in the air samples in the air chill room. However, on the carcasses post air chill here, you didn't see any red bar here, so it's not very present on the post air chill carcasses of Pseudomonas. So one of the reasons I speculate is of the processing parameters between line A and B, whereas line A is 1.5 hours, line B is half an hour. Because the less time you spend, each carcass spend in the air chill, you may potentially have less chances to redeposit the Pseudomonas onto the carcasses. And in line B, the other significant finding is this particular organism called Anoxibacillis, and that's a thermotolerant organisms, and usually you find this particular organism in the hot spring environment. And whereas in line B, we did find significant distribution across all the carcass examples and also in the environmental samples. And if we refer back to the processing or differences between line A and B, line B has a one tunnel system that incorporates scald, defeather, and placa. So the potential reason is because you got all these three steps together, and then the inside the tunnel is quite humid and warm. So it's a sort of an optimum condition for Anoxibacillus to grow and survive. And also the reason of having the scalding at high temperature is to open up the fossils on carcasses in order to remove feather in the following stage. But the downside of that is you may potentially encapsulate the organisms into the chicken skin and then carry over to the following processing stage. And then here I present the looking the diversity between different processing stages of line A. Top the graph shows the two big classes, where you have the pre-score, post-score, and Zika cluster together, and the pre-immersion, post-air, and post-immersion cluster together. And looking through the literature, some studies also find similar results. They define as this pre-immersion, post-air, post-immersion as defined as clean area during the processing, and the other three stages as dirty area, which is reasonable because between these two, after post-score, you got the feathers and intestines removed. And when we look at the 10 most abundant OTUs across sampling sites, which are altogether 28 different genera, one of the, you can see the post-air chill sample are quite separate from the rest of the samples. And that's potentially a reason. That's potentially because of the significant abundance of Pseudomonas and spoilage organisms in the air chill in line A. So it shows the contamination of the air chill during the processing. When looking at line B, generally between the all OTUs and the 10 most abundant OTUs are quite similar, because you got the wall surface samples and end of shelf life samples, which are dominated by Pseudomonas, together clustered on the other side, and then largely the carcass samples clustered in one large group. You have the pre-score separate, and the same as in the top 10 most abundant OTUs. The only difference between all OTU and 10 most abundant OTUs is just the immersion chill water and the air in the immersion in the air chill room. That in the bottom graph they cluster together, and on the top graph they are separate, which is also reasonable because usually the chicken carcasses may carry a little bit immersion chill water into the air chill room that leads to similar sort of diversity profile. And but line B here again emphasize the impact of the incorporated tunnels, largely due to the dominant presence of the Anoxibacillus. So from the two samplings, we find the culture-based results shows effective in reducing bacterial population, and evisceration air chill may cause contamination, and the Campylobacter are persistent on carcasses through the processing as 100% prevalent on carcasses. And 16S results shows deeper understanding into each processing stage, particularly that in line B, the one tunnel system may have undesirable selective impact of the thermoduric organisms such as Anoxibacillus and also the Campylobacter, which requires higher degrees compared to E. coli and Salmonella. And each step shows alteration of bacterial diversity on carcasses. And another significant finding from the line B and A is time-dependent impact of immersion and air chill may have a greater impact of the diversity on carcasses. And then look at the relative abundance of Campylobacter shows that it is a minor component in the organisms, in the bacterial community. Based on the two samplings, and then we decide that the scalding, immersion chilling, and air chilling are important processing stage through processing. And then the other part of the study is to help the industry to find better solutions that can potentially reduce the presence of the Campylobacter, because the current situation is, if I'm not sure whether any of you can try to recall and think about when you buy a chicken from supermarkets, do you actually smell some unpleasant odors? Because one of my friend in the office here in Brisbane, that he told me he can definitely tell there's residual chemicals on the chicken that when he bites. So that's because of the residual chlorine in the current commercial application. And in Australia, and the same as in the USA, application of chlorine is allowed, whereas in EU is banned, because it is believed that the chlorine is effective to reduce the foodborne pathogens. However, recent studies have doubts about that. And the other reason of the chlorine that the current Australian industry try to look for alternative is the bleaching effect. Some customers complain and says, comparing to before, the chicken they buy now, they feel that they looks a bit of a brighter and whiter. And during the processing, there's a potential human carcinogen byproducts that are caused from the reaction between chlorine and residual organic matters in the processing, and as well as the unpleasant odor of the chlorine on the chicken products. So one good candidate to replace chlorine is the prussic acid. It dissipates into water and oxygen, so it doesn't actually cause byproducts to potentially pose harm to humans. And however, in Australia, in poultry processing, the application of PA is still under consideration, and there's no clear guidelines in terms of how to implement PA. But in the horticulture and other sort of foods, the potential concentration of PA on the final products is less than 0.7. And in the USA, they've already implemented PA during poultry processing, which we can refer to. And the concentration is usually between 200 to 2,000 parts per million during chilling. And so to help the industry to find the potential application of prussic acid, I developed 3 different food matrices, refers to M1, M2, and M3. M1 is buffer peptone water, M2 is the chicken breast meat, M3 is a meat-based broth that I stomach the meat with the buffer peptone water to create a situation that's similar to the immersion chilling tank. That's where the current chlorine is being used. So if we need to move along to potentially use PA, that's a potential processing stage may be used. And in our lab, we have 17 different CJG9 isolates we collected from national surveys. So to choose potential isolates that I will use for this food matrix testing, I look at the previous results, and then I chose 2 different isolates, which is 2674 and 2704. 2674 shows the significant difference between 53 and 57, 2 different temperatures, whereas it also shows a significant resistance to chlorine. And the 2704 shows least variation between 2 different scalding temperature, and it shows a relatively less resistance to chlorine. And then I developed 8 different combinations of processing parameters. So largely the variation between depends on the different scalding temperature, and also whether the chilling or chilling with the prussic acid is involved. And so they divided into sort of a group as C1, C2 as scald alone, and then C4 and 5 is the scald with chill. C7 and C7 and 8 is the scald and chill with the prussic acid. And then you got C3 and C6 just to test and chill with the prussic acid. So here's the result in 3 different food matrices across all combinations of processing parameters. Overall, that shows the application of the different interventions on chicken meat is the least. And then one of the interesting finding is C3 is just the chilling alone. It shows absolutely no results or no impact on the CSIRO on 2 isolates. And then the other interesting result is look when we compare the C5 and C8, which is the highest scalding temperature with chill, and then C8 is the highest scalding temperature with chill and prussic acid. In buffer pathogen water and chicken breast meat, it shows significant reductions on 2 isolates. And especially between C5 and C8, the application of PA shows significant improvement in the CSIRO reduction, whereas in meat-based broth, there's no significant reduction or increase or decrease between C5 and 8. Shows that, so which indicates the organic matter in the immersion chilling tank may potentially hinder the impact of prussic acid. And the other interesting finding is a C6 in buffer pathogen water and on chicken meat. It shows greater reduction of CSIRO, even greater than the C8, which is the scalding plus chilling and prussic acid. Whereas in meat-based broth, there's no significant reduction or increase compared to the rest. So here seems like the chilling with the prussic acid is the most effective combination. And so in that, based on that, the potential application of PAA could be in the clear water, whereas is the potential of the secondary immersion chilling tank, where you have less organic matters residue, or in the air chill when you spray the prussic acid straight onto carcasses, or post air chill dip, where you have less organic matter and apply the PAA in water. And overall of the food matrices, the results it shows shows the earlier that the chill with the prussic acid is the most effective one, and then it's even effective than the combination of heat and the chill with the prussic acid. So the one potential hypothesis it poses is the prior scalding may cross-protect Campylobacter in subsequent prussic acid application. And if that's the case, we need to understand the survival mechanism, whether actually prior scalding or prior the heat resistance of Campylobacter may hinder the oxidative stress of prussic acid. To do that, I have to choose one isolate, and based on the variation between 2 isolates across combinations, earlier 2674 is the one that shows significant resistance to chlorine, whereas here under the prussic acid, 2674 is susceptible to prussic acid. 2674 is susceptible to PA, and 2704 is resistant to PA. So in the end, I choose one isolate 2704 for the following proteomics study to understand the survival mechanism, and to find whether heat resistance or heat response of CSIRO involves in the prussic acid application. In order to achieve that, I applied 60 parts per million of PA as sublethal concentration and exposed the cells for 45 minutes and extract the proteins across 4 time points as 0 as control, 15, 30, and 45 minutes. So the whole experiment involved 72 samples, including 6 bioreps, 3 tech reps, and 4 time points, and then sent for a collaborator in Brisbane within CSIRO to do the protein analysis. And the result shows here on the left is the profile, the protein profile between different time points. As you can see here, the 0 minute as control clustered as one large group, and then you have 15 minutes separated from the 30 and 45 minutes. shows the protein profile between 15 minutes are significantly different from 30 and 45 minutes. And largely among all the 109 different differentially expressed proteins across time points, largely 61 proteins are shared between 3 time points. And then the 30 and 45 minutes share another 30 proteins. And so based on the results here shows first 15 minutes, the cells may actively respond to PAA. Sorry. And then 30 and 45 minutes, the cells may already established a sort of adaptive response to prussic acid. But then by looking at the detailed protein profiles across 3 time points, a few key proteins are identified as listed here. One of the proteins called Campylobacter chaperones is usually identified as a heat response protein. However, the presence of this GRS protein in the first 15 minutes is undetected or is not significantly expressed, and in the later 15 minutes, it's actually decreased. And the other usually involved in heat resistance protein is long protease, and the presence of this particular protein is actually increased increased over time points, and so this shows the heat response proteins does involve in the prussic acid exposure. It just depends on different proteins, different heat proteins. Their role in this response may vary. And looking to the detailed function of the long proteins, it also involves it also involves the cell mobility and the biofilm formation. So the potential survival mechanism of Campylobacter to prussic acid is, instead of it may largely involve move the cells to a more favorable condition to avoid the encounter with the prussic acid. And along this, apart from the heat response, all the other sort of greater response involved the oxidative stress, ion acquisition, energy production, and cell mobility. They have a greater impact in the survival under prussic acid than heat response. And because this proteomic study is only expose the cells to prussic acid without prior scalding, and so the future study may be interesting to see whether involve prior scalding that potentially induce abundant heat proteins as normally identified in the heat response may be involved in the combination of scald and the chill. And also need to future study may need to involve to understand whether the survival mechanism is the cell mobility to move the cells to a more favorable condition, or as to form a biofilm. And from my whole project, in the end, and the suggestion, if the industries go ahead to implement prussic acid, is potentially, if they want to add prussic acid in the water, then they can add prussic acid in the secondary immersion chill tank, or the post air chill dipping tank, where you have less organic matters, or if directly apply prussic acid onto carcasses is during air chill. So this is my project, and here I would like to acknowledge and thanks all my supervisors, John, Jay, Narelle, and Leslie, and to my collaborator and advisors in both UTAS and CSIRO. And also, because I'm externally based in Brisbane rather than on campus, so I'm quite happy to have some PhD fellow students together with me in Brisbane's office, and Annalise and Jess, and I have one in Sydney. And I also like to acknowledge all the other people helped me through my PhD Intel and food program in CSIRO, and also the funding from the Strategic Innovative Program in CSIRO, as well as to thank the participant poultry processing plants in this project. Thank you, everyone. And if there's any question, I'm more than happy to ask, to answer.